All right, hello and welcome back, ALEKS students. This is another ALEKS topic that's called predicting the location in a protein of a residue sequence. And this is a really cool little biochemistry topic. I'm going to go through two examples. Basically, I'm going to start by talking about the theory behind this, talk through the strategy to work through this, and then go through a couple of examples. So basically in this topic, we're given this protein, this sketch of a protein, where we've got our N terminus here, we've got our C terminus here, and then we've got this long strand that has sort of folded up on itself and sort of formed into this protein structure here. So then sort of we would imagine that this would be sort of the protein, and then outside of the protein would sort of be all the other stuff that's going on. We've got an unlabeled and a labeled little picture here. Really, it's just to sort of show you what it would look like without these little labels in it, just sort of the big picture. This is really what we need to be worried about or focused on, though, where these little sections have been labeled for us A through G. Okay, to solve this problem, there's a few things that we need to sort of establish from the very beginning, and I've written them down here. Basically, this first part, hydrophilic residues prefer to be on the outside surface of the protein. So when we say the outside surface, that sort of means out here. So like this edge of C, this edge of E, this edge of G, this B portion here. So the outside surface is really sort of all of this stuff out here. And then hydrophobic residues prefer to be buried inside the folded protein. So like you see D here is buried inside of the protein. So D and F are completely in here. Part of A is in here as well. Sort of these ends of G and E, these ends of C and E right here, that would be considered inside the folded protein. So the take-home message there, though, in terms of like what does hydrophilic mean, what does hydrophobic mean, for our purposes, what we need to focus on is that polar means that it's going to be on the outside. So if we're looking at polar residues, then those are going to be things that want to be on the outside edge, outside surface of the protein. And if it's non-polar, we're going to find those on the inside surface right here, right? So that's going to be buried inside. So polar equals outside, non-polar equals inside. We need to remember that. Next, residue sequences are typically written from the N terminus to the C terminus. So in terms of the ordering here, this edge, like if we're reading this, for A, this edge would come first in terms of our sequence, and then A, the rest of A here are sort of as we go through, this direction, essentially, the N terminus to the C terminus. And then glycine is often found at sharp bends. So where we see these bends here or turns, typically speaking, we're going to find a glycine residue at these points, sort of at the edges where we're turning in on itself. That's going to be where we see those glycines. And you'll see sort of why that's important in just a second. Okay, so now we've got our sequence. So the first thing I want to just reiterate, and you'll notice that I've done some labeling here. We'll talk about that labeling in just a second. But this sequence, as we're reading it, going back to that second point, this we would assume is sort of at the N terminus. So like this portion here would be right here, if this was A, for instance. And then as we're reading it, we're going, through this towards the C terminus. So N terminus end here, C terminus end here. And then you'll notice that each one of these has glycine after 3 residues. 3 residues, glycine, 3 residues, glycine. Same thing for all of these. So these would indicate where these bends are happening. So 3 residues, glycine, bend, 3 residues, glycine, bend, 3 residues, so on and so forth. So that's sort of what's being, identified or sort of, captured there. So that's sort of those last 2 points. Now for the first point, this where did all my notes go? Hydrophilic prefer to be on the outside, hydrophobic prefer to be on the inside. We need to label these as polar or non-polar, and then the way that we're going to do that is from this section in the ALEKS data table. So in the ALEKS data table, we've got these naturally occurring amino acids. We have labeled all of these as either non-polar, polar, acidic, or basic. And for our purposes, all of these non-polar ones, we're going to label them as non-polar. If they're polar or acidic or basic, we can label all of these essentially as polar. They're going to be, these are going to be the things that are going to be more hydrophilic because they've got, polar groups or, acidic groups or basic groups. They're going to interact with water very well. So that's going to be, the way that we're going to label those. Okay. So then basically what I did to help us get going, and now we're going to sort of get into this, is I labeled all of these residues as either nonpolar, so NP for nonpolar. So if I found them up here, I labeled them as nonpolar. If I found them, in the polar or acidic or basic sections, then I labeled them like this. So polar, basic, acidic. Really, these are all we can consider all of these to be polar. So we don't need to really do that, but it's just sort of nice to sort of show how I'm, where I'm getting that from essentially. Once we've done all of that, so I really would, if you're doing this on your computer, excuse me, you could, just look at it. It's always a strategy, but I really would, this is a situation where I would really encourage you to go slow to go fast. So if you write this out, you take the time to, write this out, to make these notes, it will just make it so much easier. So you're not going to be making a mistake when you're going through this, in terms of finishing our learning walk here. I would just really, really recommend that. So, do what you want, but certainly that will help you go slow to go fast. Okay, so let's go ahead and do this. So for this sequence, now that I've got all these labeled, what I want to do is I want to figure out, does it fit into any of these spots? And sort of like, what would we expect? So like for A, we would need the first three residues to be polar and then nonpolar, nonpolar. For B, it would be it would be polar, polar, polar. All of these are on the outside. They're not folded inside the protein at all. So this would be polar, polar, polar. And we've got nonpolar, polar, nonpolar. C would be polar, polar, nonpolar. D and F would both be all nonpolar, right? They're all tucked inside. E would be nonpolar, polar, nonpolar. That's what we have here. Nonpolar, polar, polar. This one must be E. Notice that this is check all that apply. So if there's multiple that apply, we would certainly indicate that. But in this case, it's just going to be this E because G would be nonpolar, polar, polar, right? Okay, the next one is all nonpolar. So all of these groups, if you look them up on that ALEKS data table, they're all nonpolar. So this one's going to be D and F, both D and F, because both D and F are completely tucked inside. Both of those are going to be nonpolar. The next one is exactly the same. So if you go through, you'll notice that all of these are nonpolar as well. So this must be D and F as well. So I'm going to do one more example, to hopefully make sure that this all makes sense. Okay. And you'll notice that like once we've gone through and we know what we're supposed to be doing, the actual doing of the problem is really straightforward, right? But it's all that theory behind it that we really want to make sure that we're understanding and, we're clear on. Okay, so here we've got a good one, nonpolar, nonpolar, polar. So the ordering here is really important, right? So we really want to be looking for, okay, do we have the situation where we've got nonpolar, nonpolar, polar, and the direction is really important here, right? You might look at A, you might say, well, A has, nonpolar, nonpolar, and then polar, but we have to remember that we're going from the N terminus to the C terminus. We're going in this direction, so this does not actually fit because it's sort of backwards, essentially. We do have two nonpolars here and then a polar, but we would need it sort of to be reversed in order for this to work out. Same thing here. We're not actually going to find any spot where we see this would fit. So this is actually going to be E, none of the above. Just they don't fit anywhere essentially. Next one, we've got polar, polar, polar. So everything is polar here. So clearly that's just going to be B. All of these groups are polar. The only one of these spots where it's only going to be completely exposed to the outside, right? The outside surface of the protein is going to be B. The last one here, we're going sort of the opposite of the sequence here. So polar and then nonpolar, nonpolar. So now this one actually is going to be A because we, again, we're going from the N terminus to the C terminus. So this portion here is polar and then nonpolar, nonpolar. That's what we have here. Polar, nonpolar, nonpolar. So all the rest of the other examples that you might get, if you just follow this strategy, you should be able to figure out, which one sort of is which. So hopefully that's helpful. Thanks very much, and definitely leave a comment below if you've got any questions.